Hi, my name is Mackenzie Tade, and this is my poster presentation for my vaccine design for the SARS-CoV-2 virus, specifically the B.1.1.7 variant. As we all know, the scientific community in the last year and a half has worked incredibly hard to create safe and effective vaccines for the novel coronavirus. And so I wanted to create a vaccine for the B.1.1.7 strain specifically because it has been increasing in prevalence around the world, and it is actually more contagious and transmissible than other SARS-CoV-2 variants. This is due to the fact that it has a specific mutation at the 501st amino acid of the spike protein, where asparagine is mutated to tyrosine. And this mutation is found in the receptor binding domain of the protein, which is critical because the receptor binding domain is what controls the hinge conformation of the spike protein subunits and effectively controls receptor accessibility. So by having a mutation in that domain, the spike protein is able to bind to receptors more efficiently, which increases its transmissibility and makes it more contagious, which has led to its increased spread throughout the world. So, one of the best ways to fight novel viruses and any virus in general is by creating a vaccine. And so, as we can see in the background, these are the vaccine elements that I included in my vaccine. I started with a T7 promoter for the T7 RNA polymerase to bind and start transcription of the mRNA. I have a 5' UTR sequence, the actual mRNA spike protein, 3' UTR sequence, and then the poly A tail, which is critical to prevent degradation of the mRNA protein. And then here in the bottom right, we can actually see the wild type protein, its structure generated in PyMol. And what's different about the mRNA vaccine is that besides the 501 mutation, there are actually two proline mutations that are also put into the spike protein mRNA. And the proline mutations are found at the amino acids at 986 and 987, and they're critical because they lock the spike protein into a pre-fusion conformation. And this pre-fusion conformation is the most antigenic form of the spike protein, meaning our bodies create the most antibodies when the receptor or when the spike protein is in this conformation. And this is what the Pfizer and Moderna vaccines have been using is those two proline mutations to lock it into its pre-fusion conformation. So the aim of the study was to create an effective and safe vaccine design for the B.1.1.7 variant. And I went about this by starting with mutagenesis. I designed 2 rounds of mutagenesis. The first round involving the N501Y mutation, and you can see here in the highlighted yellow, that is the tyrosine mutation. And then the second round of mutagenesis, it had the two proline mutations together since they're right by each other. And in this domain structure, we can see the N501 mutation in the receptor binding domain, and then the two proline mutations used to lock the spike protein into its pre-fusion conformation. After mutagenesis, when we have the mutated DNA of the spike protein, we are able to clone it in a plasmid vector. And so I designed my experiment to use the PGEM3Z plasmid vector and the BAM-HI endonuclease to create or to clone the mRNA. Once we have our newly synthesized mRNA, the next design is to purify the mRNA with high performance liquid chromatography. And then after high performance liquid chromatography, when we have our finished purified mutated spike protein, as we can see here on the right, it can be used further to be introduced into a vaccine. And so on the right here, we can see the mutations in red are the N501Y mutations in the receptor binding domain. And then the green and yellow are the two proline mutations right next to each other in the spike protein. And so after we have cloned the mRNA and purified it, the next step would be to actually encase it in a lipid nanoparticle so that it can be effectively given in a vaccine, and then to actually enter that vaccine into vaccine trials to ensure that it is safe and effective for humans and that it actually does its job in stopping the spread of SARS-CoV-2 B.1.1.7. Thank you.